Hey everybody, we're going to look at Chapter 10 today. Chapter 10 is Genetic Engineering and Biotechnology. Interesting topic, somewhat of a controversial topic. We're going to take a look today at just some of the basics of what is it, and what are some of the things that are happening, what are some of the tools for some of the basic techniques that they're using. And that'll be part one. And then in part two, we'll take a look at how they're using it and some of the applications and get a better idea of kind of what's happening. So let's just go ahead and get started here. What is it? It's actually fairly broad, and there's kind of a misconception that genetic engineering is simply moving DNA from one organism to another, creating GMOs. And that's part of it, but the truth is it involves much more than that. So a lot of engineering involves just analyzation, looking at it, understanding it, sequencing it. From that standpoint, you can start to alter the DNA within an organism, or sometimes outside the organism. But in reference to just a single organism, you're kind of changing the DNA. And then the other application, as I mentioned, is where you can then take the DNA from one organism and then literally transfer it and insert it into another organism. And that is another part of genetic engineering, really a certain part called recombinant DNA technology. And we'll get into that at the very last part of this topic, and then we'll touch on that more in part two. So real quick, some context here, because what's interesting about genetic engineering is it's really just a much more specific version of what's been going on for about the last 10,000 years of humankind. So the idea is that people have been genetically modifying organisms since the earliest time period that people started settling down and growing crops and kind of having the traditional farming type of lifestyle. Since that time, people have been selectively breeding plants and animals and trying to make them a more productive and more desirable outcome over that time period, something that we still strive to do today. But the way they did it initially, and really what's still being done today, but you really just want to look at it as a more traditional version of this, is fairly simple. It's called selective breeding, and all they do is just take two organisms that they have desirable traits and selectively breed them together, just kind of like it sounds. Some of the most obvious examples that we're familiar with are things like dogs, and this goes for almost any domestic pet or animal in general: horses, pigs, chickens, cats, certainly. So all sorts of selective breeding. But to be honest, cats probably less selective breeding. Most cats just breed kind of wild, but with dogs, lots of selective breeding. And this is where you get purebreds. So, there's lots of mutts and things like that. But in terms of creating purebred dogs, that's been done through selective breeding. And again, I'm not going to go into detail. I think you understand the idea. But basically, you're just picking the traits and then matching those up. So where they would never occur in nature, you're kind of forcing that sort of crossing of genes and mixing of traits. And you get a lot of dramatically different outcomes when you do that. The difference is it takes a long time. So you're modifying the organism, by a lot of technical means. You're creating a modified organism. It just takes a lot longer, and it's a more subtle process. But that's the idea of selective breeding, and this is how you get so much of a variation in dogs, for example. And everything from a Chihuahua to a Great Dane is technically the same species. Certainly arguable that they shouldn't be classified that way, but the idea is you have that much variation just within a single species due to selective breeding. So, you can argue there that you're really, really, really significantly modifying those organisms. The same is true for plants. Let me just pull up a quick figure here. Things like corn really are a highly modified version of their ancestor plant. So here's an example where you have a corn plant, what we all know as corn, and then its kind of distant ancestor called the teosinte plant. And you can see the resemblance. It has these tiny little kernels to it, but with selective breeding, larger and larger versions of that plant were selected over many thousands of years, and now we have a result which is a much more productive, much more fruitful crop that we know as corn. So this is the idea of selective breeding, and generally considered natural, and it gives, again, all these highly modified versions of organisms. To put the modern version in context, they're doing the same kind of modification, except with a greater amount of precision and control. And the difference is that you have a lot more precision over what you're doing, as opposed to some of the random nature that comes along with selective breeding. So, for example, one of the things that happens with selective breeding is that you you pick for a trait that you see, and then you inadvertently pick for another trait that you don't see. So, for example, when people pick a trait of white, fluffy hair color like that, what they're not necessarily realizing is that that is a trait that is it may have other traits associated with it. So, for example, white is actually a recessive trait. White is a color that is seen when you have a lack of pigment. So you actually have pigment genes that produce color, and then if those become totally mutated and they don't function anymore, then you get white as a result of no pigment. These extreme white colors here. So that's what results from a recessive mutation. So when you select for that white color, you're actually picking a recessive gene. And one of the things that happens is that you tend to select for other recessive genes that you don't necessarily see. And these are things that can relate to allergies, they can relate to eye problems, and all sorts of weird traits that pop out. If you look up the genetics of any purebred dog, they all have their own kind of disorders and recessive traits that go along with them. Doesn't mean every dog will have the trait. It just means that they're more common within that breed. So certain breeds end up having more traits than others. So the idea is that all these dogs are created through a very artificial process, selective breeding. Okay, so with this next part, I just want to mention the fact that everything we're going to be learning about going forward here comes from our knowledge of how DNA works. Kind of like how TVs are the product of our understanding of the electron and computers and a lot of electronics in general. Genetic engineering is a result of the understanding of the DNA itself, and that understanding is translated into a whole new industry, really, that creates drugs, is even used in forensics and crime scene investigation, everything from ancestry to diseases, and now creating new life forms. And that's where it gets really controversial. I think it's important to recognize that the same technology goes into all these other things at the same time. So we got to be careful how much we allow, and careful that we understand the difference of one type of application to another. A lot of people hear the word genetic engineering and they immediately say, oh, that's bad. Well, let's just recognize that they're doing a lot of good things with it too. Creating new organisms like GMOs is an area where I think that does, rightfully so, get controversial. So let's talk about some of the fundamentals that go into the whole process. One of them is that you can manipulate the DNA in other aspects of the cell outside of the living context, outside of the cell. Like the DNA, for example, can be extracted from the cell and then manipulated in a certain way that allows you to bypass some of the barriers you have within the cell. So, for example, within the cell, DNA typically unwinds through the work of enzymes. You typically have enzymes like helicase is an enzyme that splits the DNA apart. You have enzymes like DNA polymerase then that come in and replicate and add to it. But the idea is there's a natural process within the cell. And normally you have an enzyme, for example, to take care of this process, like unwinding the DNA. Outside of it, though, what you can actually do is bypass that with something as simple as heating the DNA. So if you take the DNA out of the cell and you no longer have the helicase to split the DNA, one way to get around it is just to simply heat the DNA up to 94 degrees Celsius. And it's actually easier because all you have to do is apply a certain amount of heat. And outside of the cell, at that temperature, the DNA will just automatically split apart. So you can take the DNA, here's a figure, you can take the DNA that's in a double-stranded form, heat it up, and it splits apart like that. And what's even more amazing is that if you cool it down, it will actually come back together. And so by simply heating and cooling, you can separate the DNA and then join it back together with basic heating and cooling applications. I say basic, they're advanced heating and cooling, but they're still as simple as heating and cooling, fairly simple stuff. Now, why that's an advantage, and really you can't necessarily do that inside of the cell, not as easily, because if you start heating things up to 94 degrees Celsius, many organisms start to die. I mean, that's about boiling. That's close to 200 degrees Fahrenheit. So you can't necessarily do that inside of a cell, but you can if you take the DNA out. So by taking the DNA out, you actually have certain things that make that process easier. And just to clarify, it works outside of a cell similar to the way it works inside of the cell. So not only can you do it, but in certain ways it's actually easier. Okay, so here's the first review part. Go ahead and pause it, take a look at these questions, and we'll go on to the next part. So heating up the DNA is a very, very simple way to get the DNA molecule to separate. And just to clarify, that's a starting point for most applications, because if you remember from Chapter 9, the DNA has to be opened up or unzipped in order for the information to be accessed. And so when it's closed like this, you can't necessarily manipulate it. Only when you open it up can you really do the full extent of manipulation, at least. So, some things you can do when it's closed, but to get the full set of techniques to work, you have to do this at some point. So that's just a simple way to get that done. Now, other techniques have come along over the years that have allowed scientists to then go on and further manipulate DNA in a more specific way that, not everything works as simple as, heating and cooling. So, for example, one of the first discoveries that actually really set the whole field up for advancement was an enzyme called a restriction endonuclease. And this is it's often referred to as a restriction enzyme. Let me just clarify that. You hear it both ways. The textbook called it a restriction endonuclease, but you'll also hear them commonly just referred to as a restriction enzyme. I tend to go back and forth without even realizing it, to be honest. I was always taught restriction enzyme. The textbook describes it as a restriction endonuclease. I don't mean for that to be confusing. I just want you to know it's said both ways. But they're the same. It's the same thing. And what it is, it's an enzyme that cuts up DNA and essentially acts as a pair of like molecular scissors, is how I like to describe it, and that's how figures and things and other people describe it. It's basically an enzyme that gives you the ability to cut the DNA up at certain sequences. And these were actually initially discovered in microorganisms themselves. It turns out that Mother Nature has invented these as a way to ward off invading viruses. So recall that bacteria are infected by viruses called bacteriophages, and that these bacteriophages can affect the life cycle of the bacteria. So over time, the theory is that these bacteria evolved a way to kind of reduce the effect of these viruses. And one way is that they created these enzymes that will chop up certain sequences of DNA. And so there's certain sequences that if they're present, like sequences within a virus, for example, if those are present, then these enzymes will chop those pieces of DNA up and prevent them from infecting the cell. So that's the natural function. What scientists did was they discovered these, and then discovered some initial ones, and then went on to discover many, many more. Over the years, have got to where there's currently about 3,000 of them, and probably many more yet to be discovered. But they're able to cut the DNA up, as I mentioned. Let me come right back to that. Actually, here's a figure, not an actual realistic animation, obviously. But it's just a demonstration here that the enzyme has a shape that will fit around the DNA at a certain sequence. And when it binds to that sequence, then it will actually cut that DNA and separate it. Now, just to back up here, there's 3,000 different types. What's different about each one is where they cut the DNA. They cut at specific locations. So of these 3,000 different enzymes that all cut the DNA, what makes each different is the exact sequence. And what's interesting is they have specificity for nucleotide sequences. It's a very, very unique type of specificity. So we talked about lock and key specificity. Well, this is kind of stretching that concept, and it still holds true, but it gets harder to visualize it here. But the idea is that the specificity of the enzyme is actually in relation to the sequence of nucleotides. And it actually has specificity for sequences that are referred to as palindromes. Palindromes are a sequence that are the same forward as they are backward. So for example, looking at EcoR1 down here, EcoR1, by the way, is the name of the endonuclease, the restriction enzyme. Usually named for the species they're discovered in and other parts of the bacteria there. I'm not going to worry about that too much. But there is a naming system to these. That's why they get these kind of very technical acronym type names. But like EcoR1, for example, it's going to cut anywhere there's an AATT in one direction and then an AATT in the other direction. So that's called a palindrome. So that particular sequence, this enzyme has specificity for it, and when it finds it, it binds and it makes a cut in that exact area. Now, most of these make what's called a sticky end cut, and what that means is that it leaves overlap on each side of the DNA molecule. So rather than just cutting it right down the middle, which is really like what scissors would do, these scissors make cuts on different sides of that palindrome on each side of the DNA molecule. So one side is made on the front side of the palindrome, the other cut is made on the back side of the palindrome. And that creates sticky ends. And I guess the theory on why that's advantageous is it allows DNA to then join back together more easily. So by having these sticky ends, DNA can actually splice back together more easily than it could if it was cut straight across. Sometimes they do. Sometimes you have what's called a blunt end, not shown here in this figure, but occasionally there's a cut that just goes straight down, but those are not quite as common. Okay, so you have enzymes that make cuts at DNA. These are called restriction endonucleases or restriction enzymes. They come from bacteria as a means to chop up foreign DNA from things like viruses. Scientists discovered them and realized, here's a tool that we can use to cut up DNA. And just to kind of connect that back to the purpose of it today, scientists exploit these as a way to make cuts in the DNA. So one of the things you start to do when you modify DNA is you start to cut it up. And what these enzymes do rather is they act as a set of tools to make these cuts. And so what you have to do as a scientist is figure out where your gene you want to cut is, and then figure out what types of restriction enzymes are available to make that cut around that particular area. A lot of work that goes into it, a lot of specifics, but that's the idea behind that technology there. So that really was one of the more fundamental tools that came along that allowed scientists the ability to start manipulating DNA. And really before that discovery of those enzymes, there really wasn't anything called biotechnology like we know it today. All of that, the ability for scientists to start manipulating DNA really got its origins from that discovery. And since then, the field has just been growing with different discoveries and getting more and more detailed. But that was really, just to put that in context, a very fundamental discovery that kind of allowed the whole process to unfold. Now, other tools came along, and ligase was another very fundamental tool discovered very early, another big-time part of the process. A ligase is a little bit more general is the difference. Ligases don't have quite the same specificity. A ligase generally is an enzyme that will come along and seal sticky ends back together. So the sticky ends have the ability to kind of attach to each other loosely, but the ligase is what comes along and seals those back together permanently, making the DNA molecule a stable form that it wouldn't quite have without it. So you can get the sticky ends to kind of come together, but they're not fully sealed, and they don't have the strong bonds to make that DNA stick together permanently. So a ligase is what comes along and connects those. Excuse me. Reverse transcriptase is an interesting discovery that came from retroviruses. These are things like HIV and herpes viruses. These viruses actually are known to copy themselves and then insert themselves into the host DNA. And this is why viruses like HIV and herpes become permanent. And it's one of the reasons why you can't get rid of the infection. It's not the only reason, but it's part of that. They literally integrate into the host DNA and become permanent. One of the ways that they do that is they convert themselves from an RNA form into a DNA form. So let's just go real quick here, and I know you're probably sick of seeing it, but just to do it one more time here, just remember that the central dogma goes like this. We go from DNA to RNA to protein. Well, just to clarify, what we're talking about here is viruses going from RNA to DNA. They're going against that flow. So that's very, very unusual. In fact, it's one of the only examples in nature that you find where that takes place, at least as of now. That's maybe other discoveries will prove that statement to be wrong. But it's one of the few examples where that happens. So it's a very, very unique enzyme, and it comes from these viruses that will convert themselves into a DNA form and then insert themselves as a DNA form into the host DNA itself, into the host chromosomes. So I don't want to get into that. That part's fairly technical, but that's what it does. Because that doesn't naturally take place, because you don't normally go from RNA to DNA, this gives scientists the ability to do that where it would otherwise be impossible. Now, there's a lot of reasons why you want to do that, and those get fairly technical. So it just suffices to say that converting RNA back into DNA is really important, and this enzyme gives you the ability to do that. So that's another really important tool there. So here's part 2, going through the reviews, making these shorter anyway, so these will be a little bit quicker. Go ahead and pause it here, go through these review questions, and we'll go on to the next part here. Okay, so in the next part, I'm taking a look at some of the ways that they analyze the DNA. So a big part of the genetic engineering process is simply looking at the DNA and trying to figure out what types of sequences we have, and looking at it from kind of a data standpoint. There's just a lot of technical information in terms of gene sequencing. So analyzation is a big part of that whole process, and there's different ways to do it. So some of the most fundamental involve things like gel electrophoresis, and it's a way that DNA can be separated based on size, kind of a systematic way of organizing the DNA. Another way is using what's called a nucleic acid probe, or a DNA probe, and it's a way to scan the DNA looking for known sequences. And then there's another application of this same technology that's done in intact cells versus done in a test tube where DNA has been separated. So we'll talk about all three of these here. So gel electrophoresis, one of the most common techniques used is takes advantage of some of the fundamental properties of DNA. One of those being we can cut it up into different sizes, and this goes back to the restriction enzymes or the restriction endonucleases. And the idea is that when we can not only just cut the DNA, but based on the exact types of restriction endonucleases that we use, and also based on the DNA itself, the exact sequences of the DNA, which are going to be different in every organism. So based on the unique sequences, what ends up happening is that we end up cutting DNA into a variety of different sizes, some being small, some being large. And the idea is that you can then separate those DNA fragments based on their relative size. So you actually separate them in a way where you get the smallest DNA molecules in one position, and then the largest DNA molecules in another position, and then everything in between in a relative position based on size. So I've got some visuals I'll show you here in a second. So first of all, you cut the DNA up, and then what you do is you take advantage of the natural charge of the DNA. DNA has a natural negative charge to it, and that'll be a review question, by the way. So it's negatively charged. This has to do with some of the phosphate chemical groups on it. And since there's phosphates are somewhat negative, and there's a lot of these phosphate groups, so the overall charge of the DNA is a relatively strong negative. What this does then is it allows scientists to apply an opposite charge, a positive charge, and it acts like a magnet where it literally pulls the negative charge closer to the positive. So done just right, of course, it's a very technical process, but done correctly, you can actually attract the DNA with like a magnet. So what they then do, it's a very genius process. They then take that principle of different sizes, negative charge, And what they do is they put the DNA in an auger, which is really the same kind of stuff we've used in the lab all semester, kind of that jello, semi-solid type auger. And what they do is they put the DNA in these little holes in the auger, and then they draw it through there with that charge. And what ends up happening is the auger acts as like a filter, and it selectively allows the smaller molecules to move faster and the larger molecules to move slower. So the auger acts like a matrix that simply restricts their movement. So they don't just all start moving at the same speed towards the positive. They move at different rates through that gel. So the gel just kind of gives them a little bit of resistance. So what ends up happening is the smaller molecules will move faster through the gel. Since there's less drag and there's less resistance, a smaller size gives them a faster speed. And then the opposite is true of the larger molecules. There's more drag, slower speed. And it's fairly precise. You do this, and it separates everything from small to large in relation to the exact number, the exact size, rather, of the DNA itself. So, again, the technical details we're going to try and avoid, just because we're not going to be getting down to the point of where we're going to use it. We just kind of want to understand it. But the idea is you create the auger like we have in the lab, as I mentioned. You just give it a different shape is the difference. And so you create kind of this box shape out of the auger. And one of the other key things that you're doing is you're creating these little holes called wells where you're going to put the DNA. So you create your auger in a box shape, and then you put these little holes in it. And this is where you load your DNA samples. And the DNA samples have already been cut into different fragment lengths here. So here you can see the DNA. It was cut with these restriction enzymes. We get different sizes of DNA. And what we then do is take that and load each set into different wells. So this figure might be a little confusing here. So what they've done, think of this as like one DNA sample. They call it DNA for sample 3. So this whole set of sequences here is all part of sample 3. So you take all of that, load that into the well here, and then what ends up happening is the different sizes here. We have 1, 2, 3, 4 different segments from smallest to largest. And what ends up happening is then those 4 segments separate out here based on the relative size. So what would happen is that the smallest fragment here would travel furthest in the gel all the way out here. The next largest, this one here, would travel behind it. The next largest here, the largest here. All right. And this exact distance is in relation to the overall size. Now, one of the other things that you do is you run a known a control marker over here. These are called size markers, or sometimes called a DNA ladder. All this does is separate it based on relative size. So all you really know is that this is the smallest, and that this is the largest, and that these are relative sizes in between. To know exactly or approximately how big it actually is in terms of a number, you have things that are like, for example, they can be considered 10 kilobases. They measure these in kilobases, so it'd be like 10,000 nucleotides. So you might have one that's 10 kilobases, one that's 5 kilobases, 4 kilobases. So to know that size, to have that number, what you do is you run a control group over here, and these are of a known size, usually everything from 10 kilobases all the way down to 1. So you'd essentially have 10, 9, 8, 7, 6, 5, 4 known numbers over here, and then what you do is simply match these up. So if you have, let's say this is 3 kilobases here, you look, and this here is in between that 3 number. So it's not exact, by the way, and this isn't necessarily the best example, actually, but the idea is you would approximate. It doesn't give you the exact number, but if this is 3 kilobases here, anything around that same distance would be about 3 kilobases. So you could say this is a little bit smaller and this is a little bit larger. So this gives you an approximation. So anyway, that's the idea of the electrophoresis. Cut the DNA up, separate based on size. Now, the way it actually looks is it looks kind of like this. And what you do is you actually add a chemical that makes the DNA glow, and then you can actually see the DNA as a fluorescent glowing color under a certain type of microscope, or under a certain type of fluorescent light, rather. So you take a picture of it, and it glows like this here. Let's go ahead and take a quick look at a video. This is an interesting video showing an animation of the gel electrophoresis. It's relatively quick, it does have kind of a certain context to it. It's in the reference to a forensic crime scene analysis, but it'll show the technique of separating the DNA out pretty well. So here we go.
Once DNA has been extracted from a sample and a section bearing useful code such as a VNTR or STR is isolated, the next step in DNA fingerprinting is to measure each strand and count the number of repeated sections. To do this, scientists use a technique called gel electrophoresis, which uses an electric current to push strands of DNA through a slab of gel-like material. Because each bit of DNA is negatively charged and subject to an equal electric force propelling it to the positively charged side of the gel, smaller pieces move faster than larger ones. When the current is removed, the gel is photographed to reveal how far each bit has migrated. By comparing the bands produced by the DNA samples of interest with bands produced by standard samples whose sizes are already known, the precise length of each DNA fragment can be gauged.
Okay. Take these off here. So, go ahead, pause. These are just a quick review over that process.
Welcome to this video guide, intended for use alongside the third lesson in a series of resources on the PCR process. This third video will guide you in using gel electrophoresis to...
Oops, sorry about that. I didn't realize the volume was going there. Anyway, what I was saying, I'm not sure if you hear me, go ahead and pause it, take these review. We've just got a couple quick sections left here. Okay, the next part is in reference to how DNA can be used as kind of like a tracing pattern to find different unknown sequences. So DNA consists of billions and billions of nucleotides, these stretches that go on seemingly forever at the molecular level. And one of the problems then is that you've got so much genetic information that it becomes overwhelming. And what you really need is a way to find a sequence in a huge amount of DNA. It's kind of like trying to find a needle in a haystack is really what a good way to think about it. So one way scientists have come up with a solution to do that is creating what's called a gene probe. And what a gene probe does is it takes advantage of the complementarity of DNA and RNA. Recall that DNA has a complementary base pairing where adenine binds to thymine and cytosine binds to guanine. And when it comes to DNA and RNA, it's adenine and uracil, and then once again guanine and cytosine. So you have this natural affinity, complementarity that exists between nucleotides already. Well, it turns out that complementarity also takes place in reference to big stretches of DNA. So what you can do is take a known gene sequence and use it to find a matching sequence in a large segment of DNA. And where this is useful is scientists can use it to find a gene of interest. So if we have a gene, for example, for cancer, and we know that some cancers are genetic in origin or lead to a predisposition to cancer. So if you know you have a sequence of DNA that leads someone to have a higher predisposition for cancer, what you can essentially do is look for that gene using a gene probe. So you can take someone's DNA and scan it with this probe and see if they have that same matching gene. And if they do, then you can identify a risk for cancer there. So that's just one way to use it. But it's a lot like a Google search or just any search engine for that matter. You have all this information out there in all sorts of complex forms, and you have to then put in what you're looking for, a keyword, a search phrase, and the search engine scans all of this mess of data and finds you sequences that match it and then brings those forward for you. A gene probe and the technique of using those is similar in that you have a known sequence and you're using it to search for anything that matches within this large muck of in this case biological data as opposed to computer data. That's kind of the idea. Hopefully that makes sense. I've got a quick video here I'll show you. But that's the idea of a gene probe, and the process is referred to very technically as nucleic acid hybridization. So hybridization is a term that's used to describe when two pieces of DNA or a piece of DNA and RNA even match from kind of a different origin. So they're not necessarily from the two same strands, like kind of your normal DNA would complementary bind to each other. The term is kind of used to describe how you have DNA from two different sources that are similar, that they can bind, but not necessarily from the same source. So they call it hybridization. So here's a quick video showing that.
Will denature or at high temperatures, double-stranded DNA will denature or separate into single strands. When the temperature is lowered, the two strands will anneal because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also anneal. This is called hybridization, to indicate that each strand of DNA came from a different source. Regions of double-stranded DNA in which the respective single strands hybridize to one another are said to be homologous, or have similar or identical nucleotide sequences. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface such as nitrocellulose, homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various DNA probe techniques. Non-homologous DNA will not attach.
All right, so that's the idea behind that. So generally speaking, the technology is used to, like I said, find matching sequences. Now, one of the more common applications of that is a technique called the Southern blot. What they're doing with the Southern blot is generally they're going to use it to identify unknown species, or I should say, this is one way to apply this technology with a form called the Southern blot. The Southern blot really is a certain application of the steps and exactly what types of materials are used. But the idea is that one application of the technology, excuse me, I'm fumbling on my words here. One application of the technology is to use it to identify unknown organisms. So genes are often, or I should say can be, specific to one particular species or one type of organism. There are certain genes that are only found in the human species, and we have other genes that are found amongst lots of species. We share lots of DNA in common with other organisms. But the idea is that certain genes are specific to certain species. And so if you can look for the presence of a certain gene, then that can tell you about the identity of a particular microorganism. So you have these unknown microorganisms, and you can use a gene to determine what species it is, or depending on what gene you're looking for, it might tell you what type of microbe, like maybe what type of genus or what broad category. So depending on what genes you're looking for, you can identify organisms based on the characteristics of those genes. So you can look for genes that are shared amongst a broad group of organisms, or you can look for genes that are specific to one species or another. And that's kind of the idea and how it's being applied here. So here's another quick video showing that type of technology.
The Southern blot is used to verify the presence or absence of a specific nucleotide sequence in the DNA from different sources, and to identify the size of the restriction fragment that contains the sequence. In this procedure, the DNA is isolated from each source and then digested with a specific restriction enzyme. The DNA restriction fragments are then loaded onto an agarose gel and the fragments separated by electrophoresis according to size, with the smaller fragments migrating faster than larger fragments. The fragments are made visible with ethidium bromide. The DNA is then transferred from the fragile gel to a nylon filter. Next, the radioactively labeled nucleic acid probe is added. the probe binds to complementary DNA segments. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with an X-ray film. After development, the positions of the probe become visible.
So the idea is they put 3 organisms into the test. They were looking for a certain gene, and the test results indicate that that gene was found in organisms A and C, but not B. So depending on, again, what gene you were looking at, you have to know kind of something about the gene of interest. It's based on what that gene is that would tell you something about those organisms. So it might be a certain species or a certain group, like I said. So we're not going to worry about all the steps and the technical parts of it, but from a broad concept, that's the way gene probing is used to identify species or to find genes of interest within large segments of DNA. It's really a lot like finding a needle in a haystack. Now, there's different techniques and different applications. The Southern blot was one of those. The Southern blot is good. Some of the drawbacks is that it's highly expensive. It's very time-consuming. As you can see there, there's a lot of steps, and they kind of rush through those steps. So each one takes a lot of time and materials, and the process becomes time-consuming, which makes it expensive. So scientists have been looking for ways to speed up the process and amplify the outcomes and speed up productivity, basically. One way they've done that is what's called fluorescent in situ hybridization. I think I'm pronouncing that correct, in situ. The idea is that you use a gene probe, but you apply it to intact cells. When I say intact cells, you haven't extracted the DNA from the cells. You basically take a sample directly from a patient, you fixate the cells onto a slide, and then you apply the probe directly to those cells once you've kind of processed them. And what that does then is it takes all the it takes out a lot of the extra steps that go into separating the DNA from the cells, getting the DNA extracted into the test tubes, running the DNA through gel electrophoresis, all of those steps, again, time-consuming, costly. You can more or less bypass all of those and replace them with a couple of very simple, cheap, and fast steps. And that way it speeds up the process. So it doesn't necessarily work for everything, and it has a certain application, but the idea is that you can essentially do the same probing to intact cells and skip all of the extra processing. So it's called fluorescent in situ hybridization, also known as FISH. The idea is the DNA is processed and analyzed directly, and what ends up happening is you end up looking at how the DNA attaches to the chromosomes. So they actually process the DNA at a certain part during cell division and then apply it to these chromosomes, and the DNA will attach directly to the chromosome in reference to the matching sequence. So same process of hybridization based on complementary DNA sequences. So they bind, but now you're actually looking at the highlighted DNA directly on the chromosome. And so to clarify, one thing I forgot to mention here, which is important, is that when they make the probe, what they usually will do is add a tracer molecule to that probe. And what that does is it makes the probe glow. It's usually a fluorescent molecule. So wherever the probe goes, it gives off a color that can be then visualized. So it's a way to track where it's going. So that's a key part of the process I forgot to mention. So when you're looking at these, then you can actually see these little glowing tracers where the probe has bound. And you can see they've got several different probes here. Usually you run a control. It looks like the control is the blue probe here. That's to make sure that the process worked and that things are binding as they're supposed to. And then you can usually add the variable controls that you're looking to find a gene of interest. And it looks like they might even have 2 of those here in this figure. Real quick, let me show you a quick video on that. I think that'll make it a little easier to visualize here.
In situ hybridization is used to detect the physical location of a gene on an intact chromosome. In situ is Latin for in place, and in this procedure the chromosomes are adhered in place onto a slide. The most common method of in situ hybridization uses fluorescently labeled DNA probes. This technique is known as fluorescence in situ hybridization, or FISH. To perform FISH, First, cells that have been arrested in metaphase are treated to make them swell, and then they are fixed onto the surface of a slide. This also fixes their chromosomes onto the slide. Next, the slide is treated so that the chromosomal DNA is denatured into single strands. Then special DNA probes are flooded onto the slide. The DNA probes are small pieces of single-stranded DNA with a sequence from the gene of interest. Probes are able to hybridize only with their complementary sequence, which will be at the site of the gene of interest on a particular chromosome. The DNA probes are allowed to hybridize with the denatured chromosomal DNA, and any excess probes are washed away. Either the DNA probes themselves are fluorescent or they are chemically modified to allow fluorescent labels to attach to them, so that when fluorescently labeled molecules are added to the slide, some are able to bind to the probe DNA that is bound to the site of the gene on the chromosome. Excess fluorescently labeled molecules are then washed away. The slide is then viewed using a fluorescence microscope. The fluorescently labeled molecules reveal the physical location of the gene of interest.
All right, so that's the idea. And ultimately, you're tracing those genes using a cheaper, faster mechanism there. Okay, one of the last parts here, another really important technique that's used is called the polymerase chain reaction, also known as PCR. PCR is going to be similar how it starts out as a gene probe, but then it's going to have a different kind of twist to it. The idea is that you have a process where you can use what's called a primer, very similar to a probe, almost the same thing, where you'll match up to a certain sequence of DNA. And then in PCR, what you do is you match up to where your target is, and then you have another enzyme that you add that will then copy the DNA near that target sequence. So the idea of PCR is that you're basically using a technique that allows you to make copies of certain pieces of DNA. So it solves a problem in the industry, which was having limited sources of DNA itself. You have to extract the DNA from a living organism. In many cases, you have limited amounts of DNA. This is especially true in crime scene analysis when you're trying to analyze DNA. It takes a lot of DNA, actually, to do some of the more advanced applications. If you only have a couple strands of DNA, you're very, very, very limited as to what you can do with that. In fact, you can't do many things with that limited amount. So PCR was another very important process that really revolutionized the field in that it solved the problem of not having enough DNA to work with. What you could do is take what little DNA you had and then copy it, especially the key areas of interest. And a lot of times you're working with a certain segment of the DNA. You don't necessarily need all of it. So not only can you make copies, but you can then copy only what you need and then separate that from the rest of the DNA that you don't need. And that in itself is very important and useful. So you can think of it like a molecular Xerox machine. That's kind of the traditional analogy that's used here. It's very similar to the gene probe in that it starts with a molecule binding to a complementary sequence, like the needle in a haystack. The difference here is it's called a primer as opposed to a gene probe. It's a certain matter of semantics. They call it a primer because in this case what's happening is the sequence binding is going to set the initiation of the copying or the duplicating of the DNA, but isn't necessarily the whole process in itself. So it's the primer gets the copying ready. And the idea is what the primer does is it picks what part of the DNA is going to be copied. So you have all these sequences that you could copy. The primer is the designation of exactly where you're going to copy. It's like having a book and you say, I want to copy page 89. So you have to turn to page 89 to make that copy. Well, here you have a primer to determine where exactly you make that copy. The primer is similar to the gene probe in that it's an exact sequence of DNA that is complementary to another sequence, and it will bind to that with complementarity just like the probe. But again, it's going to set the stage for the next process, which is the copying. So you have priming, and then you have the copying itself. Copying takes place with an enzyme called DNA polymerase. DNA polymerase is a universal enzyme found in all living organisms, and it does the same thing in that it copies the DNA using complementary base pairing. So it moves down the DNA strand. Anywhere there's an A, it adds a T. Anywhere there's a T, it adds a A, G, C, C, G, etc. So it's the enzyme that's physically responsible for carrying out that complementarity and that copying of the DNA during replication. So when cells are naturally replicating, this is the enzyme that takes place, or that allows it to take place. What's another interesting application of technology is that it turns out you can take the enzyme out of the cell, and in a test tube, it will do the same thing as it does within the cell, done correctly. So here's another way that scientists can then manipulate the natural process outside of the cell in a test tube, and it turns out it actually can even work better in the test tube than it can in nature. One reason for that is because it turns out that DNA polymerase is actually universal. And what that means is that even though every organism is different, the DNA polymerase more or less works the same in all organisms. And the idea is that DNA replication is a very, very fundamental process of life, and all organisms have more or less conserved the same process of doing it. So you can more or less take any DNA polymerase from any organism and use it to copy the DNA in any other organism or on any other piece of DNA for that matter. So in a test tube, you can take the DNA polymerase from a bacteria, for example, and use it to copy the DNA from a human cell. And it doesn't matter that the enzyme came from a bacteria and the DNA came from a human. That's not intuitive. It could work out that it does matter. I could be telling you just as easily that it does matter and it's highly specific, but it turns out that it's not. It turns out that it's universal in this particular case. And so sometimes you have restrictions there. In this case, you really don't. So where the advantage then comes in is that it turns out that there is at least one fundamental difference in the enzyme, and that is some of them are more stable than others. If you remember from the chapter on metabolism and nutrition, enzymes like proteins in general are subject to denaturation by too much heat. And one of the problems with that is you have to heat up DNA in order to process it. If you remember, we said earlier you want to heat the DNA up to separate it. And that's one of the things you're going to be doing in the PCR. So the problem is that when you heat up the DNA to that high temperature, it tends to inhibit most forms of DNA polymerase. And that makes the process especially problematic. So you have to go through this separate stages of heating and then filtering and then add the polymerase and then cool it back down. And it was a really difficult process. What they figured out is that there are some forms of bacteria that have evolved to have a more stable DNA polymerase. So it still works the same, but it actually stays intact at higher temperatures. And this comes from bacteria that live at high temperatures. That's misspelled, by the way. There's only one L. But the idea is that you can take that heat-stable version and use it in a lab where it allows you then to heat the DNA up to higher temperatures without inhibiting the replication enzyme, DNA polymerase. So they have a special name for these. They're called Taq polymerase. There's another version called event polymerase. And these are just the heat-stable versions of these DNA polymerase enzymes. So they work the same, they copy the same, but they're heat-stable. So now you can heat them up. And that's a very, very important part of where technology became more and more accessible. Prior to this application in using these heat-stable enzymes, PCR was much more difficult and really impractical for general use. This key little invention here has made the whole process much easier and very, very straightforward. In fact, this is something that you can do with very, very little knowledge. If you can follow directions, you could carry out a process like this if you have the right technology, the right equipment. But it's so easy that this process has really advanced that field of study so that lots of people are now doing it that couldn't do it before that. Here's a quick figure showing the copying. So here's your target sequence. You heat up the DNA so it separates. You add the primer. It binds to the matching sequence. It'll do so on each side of the DNA. And then what happens is the DNA will be copied. Now, it's not showing the DNA enzyme here to copy, but what's happening is that the DNA polymerase would bind here and then copy each side of the strand here. And then what you'd end up with is an extra copy of the DNA. So this would now be copied. And then what happens is the process is repeated and the DNA is copied again. So you start by copying the DNA once. You then end up going on to make copies of your copies. And what ends up happening is over a number of cycles, the DNA is copied, and then there's copies of copies, and you end up with millions and millions of pieces of DNA after a couple hours. This figure doesn't do a good job of showing that. Let me pull up the video here, and the video does a little better job of showing that.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
All right, so after so many hours of that, and after making copies of copies of copies, you end up with, like they said, millions. So here's the review part 4, going over the gene probes and the PCR. We've got just one last quick section, and that's going to kind of set the information up for part 2, where we're going to go into that last part in more detail. So go ahead and pause it here. Take a look at these questions, and we'll go on to the last part here. So the last part, just a quick, like I said, kind of a prequel to the next part here. We're going to be talking about recombinant DNA technology. So this is where we're going to focus on the process of moving DNA from one organism to another. Now, there's a lot of terminology here that can get kind of complicated. I'm going to try and get around it as much as possible. But one of the things that's a little confusing is they use the word cloning very liberally, let's say, to the point that it gets confusing to even to people who know this stuff. It's kind of ridiculous. Typically, you think of a clone as an exact copy. Like they do this where they do what's called whole organism cloning. If you may be familiar, that one of the first organisms done was a sheep called Dolly the sheep. That was an example of what's called whole organism cloning. And they take and they make a duplicate and they insert it into an embryo, and then they raise the embryo within a surrogate mother. This is actually a little bit different. A clone in this case involves a cloning of a gene. Copying of a gene, really, and then moving it into a new organism. So the idea of a genetic clone is a little bit different than a whole organism clone, okay? I don't know why they have to make this terminology more confusing than it already is, but there it is. So the idea behind it is that involves kind of a systematic process, and without going into every detail, we'll go through kind of the major steps here. First of all, there's a lot of information that has to be known about the gene, and really years and years and years of decades of research can go into just that part there of figuring out what you're looking at, what genes do what, where they're located, what chromosome they're on, how to get them out, or how to analyze them, and that kind of one sentence part there kind of summarizes that whole process. Assuming that you can cut it out, the next step is to find the restriction enzymes necessary to cut that piece of information out, that piece of DNA out from the rest of the DNA. That's not always possible, by the way. New technology is actually making that more and more possible. I'll take that back. Initially, it was much more restricted. I should say it's actually becoming much less difficult. But traditionally, you had to find the right enzymes to cut that out. Next, you would have to then separate the DNA and then isolate it so that you have just that piece separate. So that would be done with something like the gel electrophoresis. The next thing then, and this is where it kind of takes a new leap into new territory, is you then have to use what's called a vector. And a vector in nature is something that normally transmits DNA from one source to another. And there's not very many of these. Typically, it is either some sort of bacterial plasmid or a virus, and these are going to be modified. But in nature, these naturally have the ability to transfer DNA from one source to another. If you look back in Chapter 4, we talked about plasmids. We also talked about these in Chapter 9 just recently. And it's a piece of DNA that's mobile and is transmitted from one cell to another. So in nature, bacteria, for example, use plasmids to exchange DNA. Viruses are just known to insert themselves into a host. Go back to the idea of an HIV virus or a retrovirus, a herpes virus. They will literally integrate themselves into a host DNA molecule. They do this naturally. So what genetic engineers have done, and what virologists and microbiologists in general have done, is they've figured out how to exploit that and how to take viruses, modify them, and then use them to insert DNA into a new organism. Again, very controversial. It gets very, very technical, and they're doing a lot of things that wouldn't naturally occur in nature. But that's essentially what they're doing. It is cutting the DNA out once they figure out what they want, and then using a vector then to move it into a new organism. And ultimately what they're going to be doing then is then shuttling that DNA into a new source and then integrating whatever traits from the original source into a new organism. And from there then you start to move whatever genetic traits you had into this new this really becomes a new creature, really a genetically modified organism. So interesting stuff. So in part 2, we'll look at some examples of what they're doing and kind of how that works in a little more detail here. All right. So just one last little part here, just to go over that last section there, recombinant DNA technology. I just want you to be familiar with some of these basic things here. Okay. We'll go on to part 2 next, and we'll get into some examples, like I said. Let me know if you have any questions as usual, and I will see you on the next video.